Hello everyone, how are you? Today we are going to pass a new topic. As you see from these pictures, there are some organisms which has some changes on their normal appearance. For example, this man is very hairy, snake with double head, and some abnormal plants. So all of these pictures are having the common things which call it mutations. What means mutation? This is the changes in DNA that affect genetical information. As you know, from the previous lesson we passed about the DNA, that all genes and codes of organism is stored in this DNA. What you will learn from today's lesson? You will study the theory of mutation, causes of mutagenesis, and types of mutation. And it will be better if you will know this kind of terminology: abrupt, deletion, duplication, inversion, mutation, mutagen, translocation, and sickle cell anemia. From here you can see the types of mutations. Let's firstly learn what is the mutation. So, the DNA of all living things organism consists of nucleic acid. The sequence of nucleic acid makes up genes. The different types of gene arose by process called mutation. Mutation is random. It is spontaneous. Change in the structure of DNA means that if DNA will change its structure, there can be mutations. Mutation in different ways affect on the body, as it was shown on the previous pictures. Sometimes change of nucleotide sequence is not crucial for an organism. However, it may generate new useful genes. From these sentences, you can see the easy example of the mutation. For example, the fat cat ate the rat. What will change if we will replace one of the letter in this sentence? For example, we will change the letter C. Instead, it will put H. So, the sentence will change like this. The fat hat ate the rat. How can hat eat the rat? Is the question. You see, what is the difference? In such a way, there are types of mutation which may happen in organism. It is insertion, when not suitable letters are added or has a reversible part of the letters. For example, the normal sentence is the fat cat ate the rat. The fat cat XLV, something not understandable, ate the rat. So what is the difference between these two sentences? The next part of the mutation is deletion. From the deletion you can understand that root of this word means delayed, when the genes are escaping. For example, the normal sentence is the fat cat ate the rat, but the fat ate the rat. The escape word was cat. Frame shift mutation shifts the reading frame of the genetic message so that the protein may not able to perform its function. If we will put H, the sentences will lose its meaning and letters will change its place. Deletion. During the deletion mutation, there is one H is loose. So again, sentence meaning is changing. To understand mutation, let's take a simple letters like A, B, C, D, E, F from alphabet. If the original chromosome will have this kind of state, what will happen during the deletion, duplication, inversion, and translocation? These are the types of mutation. For example, during the deletion, the B escaped, so original chromosome changed. During the duplication, B copied itself and makes not 3 letters. Instead of it will become 4 letters: A, B, B, C, D, E, F. During the inversion, the D and E reversed, so it is changed their places and reversed into 180 degrees. And C with B also flipped their places. During the translocation, the letters have almost changed their places with other letters. For example, A, B, C, J, K, L, or G, H, I, D, E, F. They have changed their places. In such a way, our genes also can change during the mutation and cause some disorders. Let's watch the video where it will explain it in your own mother language to understand clearly.
ДНК репликациясы кезінде қос тізбек бөлініп, олардың әрқайсысы қайта синтезделген тізбек үшін матрица қызметін атқарады. Бұл тимин нуклеотидіне аденин және гуанинге цитозин нуклеотиді қарама-қарсы болуы қажет екендігін білдіреді. ДНК репликациясы кезінде ДНК полимераза ферментін катализдейді. Осыған қарамастан кейде репликация кезінде жаңа түзілген тізбекке фермент комплементарлы емес нуклеотидтерді де қосады. ДНК тізбегіне нуклеотидті жұп ендірілуі немесе ДНК тізбегінен нуклеотидті жұп жойылуы репликация барысында жүзеге асуы мүмкін. Кейбір сыртқы физикалық немесе химиялық факторлардың жасушаларға немесе ағзаларға әсері мутацияның жиілігін арттыруы мүмкін. Ультракүлгін сәулелену және радиоактивті ыдырау кезінде пайда болатын сәулелену сияқты белгілі бір түрлері ең белсенді физикалық мутагенді факторлар болып табылады.
Most are neutral, for example, our eye color and birthmarks. They can be appeared because of the mutations. Some of them are harmful. For example, sickle cell anemia. It is one of the diseases which has the problem with the hemoglobin. Down syndrome. It is the syndrome which has the problem of chromosome. Normal humans have 46 chromosomes, but Down syndrome chromosomes, they are 47. But some are the beneficial, so there are some pluses from the mutation. Sickle cell anemia to malaria, they can kill, and that bad bloods will not travel in your organism. And immunity to HIV. What causes the mutation? Of course, our environment full of many chemicals. So mutations, those that result from change caused by environmental chemicals or radiation, are called induced mutations. The environmental agents that significantly increases the rate of mutation above the spontaneous rate is called mutagens. Mutation can be inherited from parent to child, and mutation can be acquired from environment. For example, the ultraviolet, X-rays, gamma rays, etc., and mistakes when DNA is copied. The chromosomal mutations can affect our health. For example, there are some syndromes. Down syndrome. The baby has this kind of appearance with the 47 chromosomes. They develop slower than normal human and they have many illnesses, very greater in their degree of intelligence. Cru-du-Chat syndrome. It is the deletion of material on fifth chromosome. You see, the chromosome will escape and they cry like cat-like. That's why it is called from French cri-du-chat. There are some gender chromosome abnormalities like Klinefelter syndrome. Normal human male or female has XX last chromosomes or XY, but this kind of syndrome, like a Klinefelter, they have XXY, XXYY, or triple XY. They are males, but they have some problems: sterility, small testicles, and breast enlargement. Super male, they have double Y chromosomes. Actually, it should be two, but gender chromosome, they have three chromosomes, and it is called super male. So they look like normal male traits, but often they are tall and thin, associated with the antisocial and behavioral problems. You can see it from these pictures. Turner syndrome, when gender chromosome is only one single chromosome. So they are females, looks like female because of the X chromosome and exception of Y chromosome. Their gender is female. Super female, when they have trisomy X chromosomes, they are little, not visible differences, and of course they also limited fertility.
Орақ пішінді жасушалық анемияны алғаш рет 1904 жылы американдық дәрігер Джеймс Херрик сипаттап берген. Ол кезде әлі белгісіз осы аурумен ауыратын қара нәсілді студенттің қанына талдау жасаған кезде ол былай деп жазды: Ерекше таңғалдыратыны бұл ұзартылған орақ тәрізді эритроциттердің мөлшерінің көп болуы. Бұл ауру қанның қызыл түйіршіктерінің құрылысындағы қандай да бір белгісіз факторлардан туындауы мүмкін. Тек 1949 жылы ғана Линус Полинг бұл аурудың бір аллельде болатын өзгеріске байланысты туындайтынын дәлелдеді. Бүгінде біз бұл ауру гемоглобиннің ақуыздық бөлігі Б глобин тізбегін кодтайтын гендегі нүктелік мутациядан туындайтынын білеміз. Бұл жағдайда аденин тиминмен алмастырылып, нәтижесінде тек бір амин қышқылдың өзгерісіне алып келеді. Гемоглобиннің 6 қатарындағы глутамин қышқылы валинмен алмастырылады. Осындай кішкентай өзгеріс барлық қан түйіршіктерінің құрылысын айтарлықтай өзгертіп жіберді.
Please keep your health, keep yourself. Thank you for your attention. Goodbye.